Okay, so welcome to part 2 of the chemistry lecture. This lecture is going to be focused primarily on organic chemistry, just how that interacts with the body and how carbon-based or things occur. Alright. So carbon is a really important part of organic chemistry. All organic molecules contain carbon. So the carbon that organic compounds always are going to contain is going to have several properties that are going to make it particularly useful or helpful to various living organisms. So it can react with one or several other carbon atoms to form large molecules of these different, that they're going to have various kinds of shapes. And so many carbon compounds are also going to dissolve fairly easily in water. They do not dissolve easily in water. And this makes them useful materials for building body structures. If you lived underwater, you wouldn't want to build a house out of salt because it would dissolve. The same thing goes with cells and structures in the body. If you're constantly surrounded by water, you wouldn't want to have something that is fairly water soluble. So carbon-based compounds are really handy in that regard. Carbon compounds are either mostly or entirely held together by covalent bonds. And these are going to be able to decompose fairly easily, but when they do so, they're going to release a fair amount of energy. So this means that organic compounds are going to be an excellent source of energy. So we can break bonds, and that'll release energy that can be used for whatever bodily processes we need. So the chain of carbon atoms in an organic molecule is called a carbon skeleton. And this is going to be, and attached to this carbon skeleton are going to be these very distinct functional groups. And there are lots of different functional groups, but these functional groups are going to be other elements that are bound to the carbon and hydrogen atoms. And this is what makes them unique, and this is what gives them their functionality. So we had this crazy big table. You can see it's 3 slides long of all the various functional groups for organic compounds. Now, I'm not going to ask you to memorize all of the structures and the functions of these functional groups. Just suffice it to say that on a carbon skeleton, you can have any of these functional groups added to that carbon skeleton to give it different functions. So things like alcohols, this is going to contain an OH, a hydroxyl group, or you can see there. So R is, well, I don't know what it really stands for, but R, in my mind, just stands for random thing that it's attached to. A lot of times it'll be part of the carbon skeleton that it's attached to, but this is just whatever is attached to. All right, so this is just indicating that this OH group, this hydroxyl group, is going to be attached to something else. All right, this is part of what makes it an alcohol. All right, so anyhow, in this instance here, this alcohol, this is going to contain an OH group, and this is going to be a polar and hydrophilic sort of functional group, and this is due to that electronegativity of that oxygen molecule, of that oxygen atom. And we talked about electronegativity in the last couple of videos, where that really big oxygen is going to kind of hog the attention of those electrons that are swirling around, that it's sharing with that hydrogen. So these molecules that have lots of these OH groups, these little alcohol groups, are going to dissolve fairly easily in water. A couple of other functional groups that we have the sulfhydryls. These are things like thiols. This is a sulfur hydrogen, sulfhydryl group, right? These are also called thiols. They have this sulfur, this hydrogen bound to them. These are hugely stinky. They just smell like rotten cabbage or something. They're terribly stinky. But yeah, so these are just some of the functional groups that can be added on to those carbon backbones, those carbon skeletons, to give them very distinct functions there. Right? So we have ketones. These are going to contain a carbonyl group within the carbon skeleton. So the carbonyl group is going to be polar and hydrophilic due to the electronegativity of that oxygen atom that we see here up at the top. Another carbonyl that we have is the aldehydes. We have the ketones and the aldehydes. So aldehydes are going to have a carbonyl group at the end of the carbon skeleton. So you can see here, this is where you would attach, that R is where you'd attach to whatever random carbon skeleton we'd have. Our carboxylic acids are going to contain a carboxylic group at the end of the carbon skeleton. And so once we talk about amino acids, like the building blocks for proteins, so all amino acids are going to have this carboxyl group at one end of the amino acid. So the negatively charged form is going to be predominant in the pH at physiological pH. And this is also going to be a hydrophilic kind of molecule. Let's see. We then also have our aminos down here. This is another functional group that's going to be found on all amino acids. So this has this amine group. This amino is going to have this nitrogen with the two hydrogens bound to it. So an NH2 group. This can act as a base, and it's really great at picking up hydrogen ions. And this is going to give the amino group a positive charge. So at the pH of bodily fluids, so physiological pH, most amino groups are going to have a charge of +1. And also all amino acids are going to have an amino group at one end, like I mentioned. Okay, so with organic molecules, like we said, they all have this kind of carbon backbone here, and there are multiple ways that we can illustrate or write out these the chemical formulas or the structure for these organic molecules. So a lot of the times, if you were to write out all of the atoms in a glucose molecule, in this example, you could see all of that is written out with all the various carbons at each of the carbon skeleton parts. With all the little functional groups added onto them. However, a lot of the times, for just brevity's sake, what people will do is they will use a standard shorthand where they just sort of will just omit the carbons there, and so they just won't draw them. And so any kind of point there is going to indicate that the carbon is there, but it's just not illustrated. So as far as the hydroxyl groups, remember that's an OH group, all right? So we want to look at how many hydroxyls does a molecule of glucose have, so we can count them, all right? So we can go 1, 2, 3, 4, 5. So this particular, unless I'm missing some, 1, 2, 3, 4, 5. Yeah, so this molecule of glucose is going to have 5 hydroxyl groups, all right? And then we can count also the number of carbons that are going to be part of the carbon skeleton, all right? And so the carbon skeleton is going to be this main sort of this six-sided structure here, right? So we're going to have 1 carbon there, 2, 3, 4, 5, all right? Because this last point there is going to be taken up by an oxygen. So there's 5 carbons in the carbon skeleton for glucose, all right? So, organic molecules. These are really important for just all of life, essentially. So small organic molecules are going to be able to combine to form very large organic molecules. And these are either called macromolecules or polymers. And they're called polymers when they're composed of these repeating units of monomers. So if we had a whole bunch of glucose molecules, that a glucose molecule would be a monomer. And if we had a bunch of repeating glucoses, that would be a polymer, all right? If we were to mix together a bunch of different monomers, that would end up with a macromolecule, right? So when 2 monomers join together, the reaction is usually done via dehydration synthesis, all right? So remember, the removal of some sort of water to form a bond, all right? And macromolecules are going to break down into monomers, typically via hydrolysis, right? So the adding of water to cleave some sort of bond, right? Hydrolysis, cleaving or cutting with water. So molecules that have the same molecular formula but have different structures are going to be called isomers, all right? And we see that here with glucose and fructose, right? The chemical formula or the molecular formula of both glucose and fructose is C6H12O6, right? But we can see here that structurally they look very different. This is a 5-membered ring, while this is a 6-membered ring. So they're very different looking molecules, but they still have the same molecular formula. So these are isomers, all right? Okay, so now moving forward then to my favorite of the organic chemistry molecules, the carbohydrates. These are very tasty in my opinion. So carbohydrates are going to provide a lot of the energy that we need for life to occur. This is going to include sugars, starches, glycogens, cellulose, if you're a plant. These are all carbohydrates. So some carbohydrates are going to be converted to other substances, which are used to build, which are used to build structures and to generate ATP. So ATP is like the main energy molecule in the body. So carbohydrates are converted into other structures to help generate ATP. So other carbohydrates are going to function as food reserves. So the general structural rule for carbohydrates is 1 carbon atom for each water molecule, all right? So it looks like this. So we have CH2O, 1 carbon for each water molecule, all right? One C and 1 H2O. So carbohydrates are divided into 3 major groups based on their size. We have monosaccharides, disaccharides, and polysaccharides, all right? Okay, so here we have just a cute little table that really nicely illustrates the examples and the definitions or the types of our types of carbohydrates. All right, so here we can see we've got our monosaccharides. These are just basically simple sugars that contain 3 to 7 carbon atoms. This is your stuff like your glucose, your fructose, your galactose, deoxyribose and ribose, things that like make up your DNA and your RNA. All right. Next, we have our disaccharides. These are simple sugars that are formed from the combination of 2 of these monosaccharides up here, and they're formed by this dehydration synthesis. Okay. So sucrose, like just regular old table sugar, like the stuff that you put in your coffee in the morning, probably, is a combination of glucose and fructose, and these 2 things come together via that dehydration synthesis. All right, and then last but not least, we have our polysaccharides. These are formed from tens to hundreds of these monosaccharides, and they're joined again by dehydration synthesis. This is where we talk about our glycogens and our starches, those really energy dense sort of carbohydrates. These are like your whole grain breads and stuff. And then if you're a plant, you have cellulose. This is your polysaccharide for plants. This is a handy kind of carbohydrate, but it's one of those weird ones that humans really can't digest, so we can't really break it down, so it makes for an excellent source of like fiber and roughage. So like this is why it's important to like eat your salads and stuff. Good fiber moving through those intestines. All right, so our monosaccharides and our disaccharides are really important sources of energy. So monosaccharides are going to contain, like we said, anywhere from 3 to 7 carbon atoms, and this is going to include glucose, which is a hexose that is the main energy supplying compound in the body. Right? So we need glucose basically for any kind of functional activity in the body, cellularly speaking. Disaccharides are going to be formed from 2 monosaccharides via dehydration synthesis, and they can split back into those simple sugars via hydrolysis, and so things like glucose and fructose are going to combine to produce sucrose. All right, and if you wanted to split them back into their individual parts, their glucose and their fructose, you would just simply need to perform some sort of hydrolysis reaction. All right. So then polysaccharides, like we said, are going to be the largest of those carbohydrates, and they are going to contain hundreds of those little monosaccharides all linked together. So the principal polysaccharide that we find in the human body is going to be glycogen, and this is going to be stored primarily in the liver, as well as in the skeletal muscles. So starches are going to be polysaccharides from glucose in plants, and then cellulose is a polysaccharide that's formed from glucose, but it can't be digested by humans. So like I said, great source of fiber, clears out those intestines, but not super, energy dense for us as people. So our next type of organic molecule or organic compound that we're going to discuss is lipids. This is another very tasty organic compound, in my opinion. Everybody loves some fat. So lipids, just like carbohydrates, are going to contain carbon, hydrogen, and oxygen. But unlike carbohydrates, they're going to have a bit of a different ratio. So they're going to have 2-to-1 ratio of hydrogen to oxygen. All right, so it's just slightly different there. So they are going to have fewer polar covalent bonds and are therefore going to be mostly generally getting ahead of myself. So they have fewer polar covalent bonds and are therefore going to be typically insoluble in polar solvents like water. This is, again, where you think of lipids like oil, which lipids are an oil. An oil is a lipid. Lipids or oil doesn't mix with water, which is a non-polar which is a polar solvent, all right, like water. So oil and water don't mix because oil, which is a lipid, is going to be very hydrophobic. So to become soluble, they have to join with these hydrophilic proteins in order to become lipoproteins. And the way that works is there's a protein, essentially, that's attached to this lipid molecule, and that protein molecule is basically going to wrap around the lipid part of the lipoprotein, right? So that the hydrophobic lipid is on the inside, and our hydrophilic protein, our water-loving protein, is on the outside, right? So this basically is a win-win situation because the protein gets to interact with the water, and the lipid gets protected and shielded by that protein, all right? So that allows it to become a little bit more soluble. So lipoproteins are going to be soluble because the protein on that outside is hydrophilic, and the lipid, on the inside is hydrophobic. So everybody kind of works together. So there are several different types of lipids that can be found in the body. Fatty acids are a big one. These are going to be used to synthesize triglycerides as well as phospholipids, or they can be broken down or catabolized to generate adenosine triphosphate. Remember, ATP. That's the main energy molecule that we use in the body, all right? So ATP is a very important molecule, so keep that in mind for later. Another type of lipid that we find in the body is triglycerides. These are our fats and our oils. These are great for protection, insulation, energy storage. These are the things that, like the fat pockets behind the eyes, that keep everything cushioned and where they're supposed to be. This is also, if you get a little chunky around the middle like myself, this is just an abundance of triglyceride, but it's great for energy storage. So if ever there is some sort of crazy famine, if you've got a little extra triglycerides hanging around your midsection, you'll be okay. Next, we then have phospholipids. These are a major lipid component of the cell membrane. So we'll talk about this more in class when we talk about the cell biology kind of lecture. But phospholipids are very important for forming and that barrier and that membrane that's going to hold the cell together. We then also have steroids, and these are actually lipid-based. We have a whole bunch of different types of steroids here. The big one that people like to talk about, especially in the medical field, is cholesterol. And so this is going to be a minor component in all animal cell membranes, and so it does provide some rigidity to that cell membrane. This is also a precursor to bile salts, which are really important for digesting food. It's also involved in vitamin D production as well as steroid hormone production. So steroids are important. They're not just for athletes looking to bulk up. So we then also have something called eicosanoids. We're not going to talk too much about this, but these are just basically a very diverse group of lipids that are going to have an effect on the body in modifying responses to hormones as well as in blood clotting, They're important in inflammation and immunity. So eicosanoids are very highly varied. They do a lot of different things. We're not going to get too much into that. And then we have our other lipids, our carotenoids. This is stuff like vitamin A. This is used to make the visual pigments in the eye. So, it's important to have a good source or a good amount of vitamin A to maintain eye health. They also are going to be hugely functional as antioxidants to kind of bind up any of those crazy free radicals that can sometimes cause cancer. So lipids do lots of things. And here we've got some more options, or more examples of lipids here. Anyhow, so the next thing that we're going to talk about is fatty acids. So these can be saturated or unsaturated. So I'm sure you've probably heard about saturated or unsaturated fatty acids, like in health food circles or like on the back of your cereal box or whatever. And so basically the saturated versus the unsaturated is basically just if everything is lined up straight and everything is saturated, if all those carbons are saturated with hydrogens, they're not going to kink. If, however, we have an unsaturated carbon, it's not full up, it's not bound completely with hydrogens, it's going to form a double bond with the carbon next to it, and that is going to cause the tail of our fatty acid to kink like that. It's going to bend. Whoop. So, anyhow, these fatty acids are used to form triglycerides, and they also are going to provide a great source of cellular energy. So like I said, they can be saturated with only single covalent bonds between those carbons and those hydrogens. This is going to be a very straight kind of tailed fatty acid, or we can have our unsaturated, or excuse me, or they can be they can be unsaturated, where there's like that empty hydrogen there. The carbon is not completely hydrated or saturated with hydrogens, and so a double bond forms, like I said, between those two carbons, and that causes a kink in the tail. So triglycerides are going to be the most plentiful of the lipids in the body, and they also provide really great protection, insulation, and they're also a fantastic source of energy. And they are also, this energy storage is both immediate, it's available on demand, or it can be stored later. In my case, it gets stored on my butt, my ass. Getting old is tough. But anyhow, these triglycerides, great source of insulation and stored energy. All right. So at room temperature, triglycerides may be either solids as a fat, like lard, or they can be liquid like oils. So triglycerides are also going to provide more than twice as much energy per gram as either carbohydrates or proteins. So triglycerides are a very energy dense kind of energy source. They're very energy dense. So triglyceride storage is also virtually unlimited. So you can store a lot of triglycerides in those little adipocytes that are responsible for storing your fat, if you will. You can always grow more of those, unfortunately. So excess dietary carbohydrates, proteins, fats, and oils, etc., are then going to be deposited in those adipose tissues as triglycerides. So in graduate school, one of my professors used to say that carbohydrates will make you fat, but fats won't make you sweet. So basically, these carbohydrates, proteins, etc., can all be converted into triglycerides. But triglycerides can never go the other way and be stored or used as carbohydrates. So anyhow, the triglycerides are going to be composed of a glycerol, which we see up here, and then our fatty acid tails, which we see down here. And like I said, these can be either saturated or unsaturated. Here we get the little kink in the tail. So the types of covalent bonds, and by inference the number of hydrogen atoms that we'd see in these tails that are found in most of these fatty acids are going to determine whether a triglyceride is saturated, monounsaturated, or polyunsaturated. All right, so here we can see both of these two tails are completely saturated with hydrogen atoms. So they are both they're both saturated. All right, here we see then a monounsaturated triglyceride because it only has one kink in this tail. If we were to have multiple places where this carbon was not fully saturated with hydrogen atoms, it would cause multiple kinks, and that would be a polyunsaturated triglyceride there, all right, or polyunsaturated fatty acid there. So that brings us into our phospholipids. These are going to be really important in cell membrane components, and these guys are amphipathic, meaning they have both a polar and a non-polar region. All right. So here we can see our phosphate group that's on that hydrophilic head, which is our phosphate group here. So this is our water-loving part of our phospholipid. This is the phosphate group. And then we have our hydrophobic lipid tail here. All right. So these things get together to form our phospholipid. You can see the polar head, which is hydrophilic, and then our hydrophobic tail. And so the way this works is that in the cell, we have this phospholipid bilayer. All right. So we have two layers of these phospholipids where they line up, and the heads all get together, and they are exposed to the liquids and the fluids that are around the cell, and they protect this inner layer of these lipid tails. All right. So these are really important for the formation of the cell membrane, which is a really big deal. Next we have our steroids. This is another type of lipid. These are a steroid by definition is going to just simply be a four rings of carbon atoms, which you can see here, 1, 2, 3, 4. This is just basically what defines a steroid. If it has this structure, this four-ring structure in it, then it's going to get classified as a steroid. So steroids can range widely in their function. They include things like sex hormones, like estrogen, testosterone. They can also be things like cholesterol with your Cheerios. Important to eat your Cheerios to keep your cholesterol under control. Cholesterol also is really important. It serves an important role in the component of a cell membrane, as well as the starting material for the synthesis of any other kind of steroids. So cholesterol is important, despite what it's important to have cholesterol in the right amounts. All right. So next we have then our proteins. Right. This is our next kind of organic molecule. We've probably all heard about proteins. Your parents maybe said to eat your meat or your chicken or whatever when you were a little kid. You got to have protein to grow big and strong. They were kind of right. That's kind of true. So proteins are going to give structure to the body. They're also going to regulate processes, provide protection, assist in muscular contraction, transport substances, as well as serve as enzymes. All right. So they do a whole lot of different things. So structurally, proteins are going to form the structural framework for various parts of the body. That makes sense. We need proteins to build muscles, basically. As far as regulatorily speaking, the proteins can also function as hormones that are going to help to regulate the various physiological processes in the body. This can be anything from control of growth and development to neurotransmitters in the nervous system, things like hormones, like insulin, all of these are proteins. So proteins have a huge regulatory role in the body. Additionally, proteins are involved they have a contractile function. They allow the shortening of muscle cells, which is then going to be important for producing movement. So things like myosin and actin, which we'll talk about when we talk about the muscles. Those are all really great examples of proteins that are involved in movement. Immunological functions of proteins. These guys are going to aid in responses that are going to be important in protecting the body against foreign substances and pathogens, things like germs, viruses, etc. All of these immunological responses are basically proteins. Additionally, proteins are great at transport, or at least some of them are. So they're going to carry really important substances throughout the body. And then last but not least, we have catalytics. All right. So proteins, we talked about catalysts in the previous lecture. We talked about catalysts in the previous lecture. You need a catalyst to reduce that activation energy for some sort of chemical reaction to occur. Well, in the body, these catalysts are proteins, and these proteins are called enzymes. Right? So enzymes are basically biological catalysts that are responsible for regulating biochemical reactions. Okay, now that brings us then to amino acids. All right, these are super important. When we talk about proteins, you can't not talk about amino acids because amino acids are the building blocks of proteins. All right, so proteins are formed by combining various amino acids together in these long chains to produce a big chunky fat protein that does whatever function in the body that it needs to do. So amino acids are going to contain carbon, hydrogen, oxygen, as well as nitrogen in this little amine group here, all right, or amino group. They're also going to contain that carboxyl group, all right. So this amino group is why we call them amino acids. All right, so an amino acid is going to, remember, it's important to remember that an amino acid has both an amino group and a carboxylic group. All right. So the way that we join our amino acids together is via a peptide bond. And so amino acids are joined together in this kind of stepwise, excuse me, in this sort of stepwise fashion in which a covalent bond is going to join one amino acid to the next in order to form something called a peptide bond. And we're really not going to get into what constitutes a peptide bond. That's a lecture for some other teacher. But anyhow, it's important to note that this peptide bond is what is joining those different amino acids together to form our resulting polypeptide chain to produce a protein. All right? Okay, so next then we need to talk about the structural organization for a protein. All right, and just like everything else, there are various levels of structural organization for these proteins. All right, we have our primary, secondary, tertiary, and quaternary. All right, so our first, second, third, and fourth structural organizational groups. All right. So the resulting shape of a protein is greatly going to be influenced by its ability to recognize and bind to other molecules, all right? And that is where that tertiary and quaternary groups come in, all right? And so then the denaturation of a protein by any kind of hostile environment like heat or acid is going to cause a loss of that characteristic shape and function. So basically, if you heat a protein, like an egg, it's going to have a significant change in shape and function, all right? So, looking at then our structural organization of a protein, all right? Our primary structure is basically just going to be a list of amino acid sequence, all right? So this is just if we went down our little protein and read off all of the amino acids that made up that protein, that would be our primary structure, all right? Secondary structure then is going to be the twisting and folding of those neighboring amino acids that are going to be stabilized by hydrogen bonds that are going to form between our various amino acids, all right? Then our tertiary structure then is going to be our three-dimensional shape of our polypeptide chain. So we've got these crazy alpha helices and beta pleats that are formed in our secondary structure, and then the accumulated function of our alpha helixes and beta pleated sheets are going to result in this tertiary structure. All right. Our quaternary structure then is going to be the arrangement of two or more polypeptide chains coming together to make our one big final protein. Right. So if for some reason we added a large amount of heat, this would cause our tertiary, our quaternary, and our secondary, and our quaternary, our tertiary, and our secondary structure to change, right? It would unravel, or it would do different things. It would form different bonds, all right? And so that functionally, that would cause a huge shift in what that protein was able to do. It's just like when you cook an egg. It starts off as this kind of liquidy blob, and then when you add heat, we have the denaturation of that protein that causes changes in the characteristic shape and function of that protein, all right, causing it to become more solid. It's way more tasty to eat, though, once you've denatured your egg proteins. So that brings us into enzymes, all right. Last time we talked about catalysts, all right. So in the biological world, a catalyst is basically an enzyme, all right. So a catalyst in a living cell is what we call an enzyme, all right. So the name of these enzymes is usually going to have the suffix ase, all right. So oxidase, kinase, lipase, blah, 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 ase. So a lipase is going to cleave lipids, all right. An oxidase is going to cleave off oxygen molecules off of some sort of molecule, and a kinase is going to cleave off some sort of protein part. So anyhow, all of this is cleaving, ase. But anyhow, all of these things are acting as enzymes. They're helping to lower the activation energy that's needed to cause some sort of chemical reaction to occur to produce energy a lot of times. So although enzymes catalyze selected reactions, they do so with great efficiency and with a whole lot of built-in controls. Otherwise, things would get out of control very quickly. All right. So the three main things that you need to remember about an enzyme, all right, is the fact that enzymes are highly specific in terms of the substrate with which they will react. Right? If the substrate ain't right, it ain't happening. All right. That substrate has to fit that enzyme perfectly. Otherwise, that reaction is not going to occur. All right. The second thing about enzymes is that they are extremely efficient in terms of the number of substrate molecules with which they will react. All right. If a single substrate molecule is present, that enzyme is going to make a reaction occur with that single substrate molecule. All right. And then last but not least is that enzymes are subject to a great deal of cellular controls. This is really important. Otherwise, if that was not the case, then we'd have just reactions running around uncontrolled and unchecked. So we need this huge amount of cellular control to determine how much of those enzymes are available to allow these chemical reactions to occur. You can't just have things running around unchecked. So enzymes are going to speed up chemical reactions by increasing the frequency of collisions. All right, they do this by lowering the activation energy and properly orienting those colliding molecules. All right, so they're getting it in the right position, and then they're lowering the activation energy that is needed for that reaction to occur, and they're increasing the amount of times that things are bonking together, smacking into each other, so that collisions can occur, so that those bonds can be broken. Right? Enzymes are very important. All right. That then brings us to nucleic acids. All right. It's kind of different, changing directions here just slightly. All right. So nucleic acids are going to be these big chunky organic molecules that are going to contain carbon, hydrogen, oxygen, nitrogen, phosphorus, etc. All right. And nucleotides are going to come in two main varieties. All right. We have our deoxyribonucleic acids. This is what makes up DNA. And this is going to form the inherited genetic material that's inside all humans. All right, so each gene is a segment of DNA molecule and thereby is responsible for regulating most of the activities that take place in our cells throughout our lifetime. All right. Then we have our ribonucleic acids. All right, this is what makes up our RNA. These are going to be responsible for relaying instructions from the genes in the cell, in the cell's nucleus, to guide each cell's assembly of the amino acids to into protein synthesis by the ribosome. All right. So DNA makes RNA, makes protein. All right. That is the biggest thing that is in cell biology. That is the central dogma. All right. DNA makes RNA, makes protein. Okay. So DNA tells the cell what kind of RNA to produce, right? And the RNA tells the cell what kind of protein to produce, all right? It only goes in one direction, DNA to RNA to protein. This is a super important concept for just cell biology and science in general. So, anyhow, but nucleic acids, these are important, all right? So we have two different types. So, yeah, never mind. Getting ahead of myself. So, a nucleic acid is basically just going to be a chain of repeating monomers called nucleotides. And each nucleotide of DNA is made up of 3 different parts. All right, we've got our nitrogenous base, which is one of 5 nitrogenous bases here. So DNA is, 4, technically. DNA is going to contain 4 nitrogenous bases, which are all going to contain carbon, hydrogen, oxygen, and nitrogen. All right. So in DNA, the 4 nitrogenous bases are going to be adenine, thymine, cytosine, and guanine. All right. So adenine and guanine are larger, and they're going to be double ringed. All right. These bases are called purines. All right. So the way I was taught to remember this is that God, guanine, G and A, guanine and adenine. So God and angels are pure. They're purines. All right. And the other 3, or 2 or 3, are going to be pyrimidines. All right. This is thymine and cytosine. All right. And then when we talk about the nitrogenous bases for RNA, we've got uracil also. Anyhow, but these 3 are all pyrimidines. All right. They're smaller. They just have 1 ring. Right, these are all pyrimidines, and our adenine and our guanine are purines. All right. So the other part of our nucleic acid, other than our nitrogenous bases, are going to be these pentose sugars. So the 5-carbon sugar called the deoxyribose is going to attach to each base in the DNA. And then the phosphate group, which we see up here, is going to be our phosphate group. Like we said, our PO4 with a -3 charge. And these are going to alternate with the pentose sugars to form the backbone of that DNA strand. And the bases are going to project inward from that backbone chain. Okay, so those are our 3 main components of the nucleic acid. All right. So nucleic acids are what is going to make up DNA. All right. The basic units of nucleic acids are going to be the nucleotides, which are composed of those nitrogenous bases, that pentose sugar, and a phosphate group, like we talked about. All right. You can see that here. All right. And then a DNA is going to be made up of 2 strands that are twisted together in a spiral staircase structure that's called a double helix. So each strand is going to consist of nucleotides that are bound together. Each nucleotide is going to consist of a deoxyribose sugar that's bound to a phosphate group, and then one of those 4 nitrogenous bases. And those nitrogenous bases are going to pair together through hydrogen bonds to form the steps of the double helix. And then always, the way that it works always is going to be adenine is going to pair with thymine, and guanine is going to pair with cytosine. Right? So A and T are always going to pair together, and C and G are always going to pair together. All right? So adenine always pairs with thymine, and guanine always pairs with cytosine. All right? So we've talked about DNA. So what about RNA? It's pretty similar, but there are some major differences here. All right? So we have, for our nitrogenous bases, we still have adenine, cytosine, and guanosine that are going to be the same throughout both. However, in DNA, our 4th nitrogenous base is thymine, while in RNA, our fourth nitrogenous base, is uracil. All right. So sugars, the sugar of that mononucleotide is going to be deoxyribose in DNA, hence the deoxyribonucleic acid. And in RNA, it's ribonucleic acid. All right. So it's going to be, that sugar is going to be a ribose instead of a deoxyribose. RNA is going to have one strand instead of two. So DNA, like we said, is going to have that double-stranded double helix, that two-stranded double helix, while RNA is going to be a single strand. So with the nitrogenous bases pairing, the same thing is going to be with the guanine and the cytosine. Those are going to stay the same. They're still, G and C are pairing together. In DNA, we said that adenine and thymine pair, all right? But in RNA, adenine is going to pair with uracil, all right? So instead of the T, it's going to be a U, all right? And functionally, there are going to be some differences here too. So DNA is going to encode the information that makes proteins, and RNA is going to carry the genetic code, and it's going to assist in the making of those proteins. So DNA codes for the production of RNA, and RNA codes for the production of proteins, basically, all right? So that is some very important differences. This is an important table. I would definitely make sure that you know the material on this table for the exam, all right? So that brings us then to ATP. We've mentioned this a few times throughout the lectures, ATP, adenosine triphosphate. All right, this is our major energy molecule for any kind of biological function. All right, so adenosine triphosphate, aka ATP, is going to be the principal energy storing molecule in the body. So among the cellular activities for which ATP provides energy are going to be things like muscle contractions, the chromosome movements during cell division, as well as cytoplasmic movement within the cell, so anything moving around within the cytoplasm in the cell, that's all powered by ATP. ATP also is going to provide energy for membrane transport as well as synthesis reactions, all right? So ATP is going to consist of 3 phosphate groups that are going to be attached to the adenosine unit. So that would be our adenosine triphosphate, our 3 phosphates, triphosphate, plus their adenosine. All right, so that brings us to ATP, adenosine triphosphate, 3 phosphates. All right, and so ATP consists of those 3 phosphate groups that are attached to the adenosine unit that are going to be composed of adenine and a 5-carbon sugar ribose. All right. So when energy is liberated from ATP, it is going to be decomposed to adenosine diphosphate plus 1 phosphorus. All right. So when we break down ATP, all right, we end up with ADP, a diphosphate. So we're kicking off basically one of these phosphates. All right, so we're going from triphosphate to diphosphate. So we're going from 3 to 1, or 3 to 2, plus this phosphate group here, which is floating around doing its own thing to provide energy, basically. And ATP is going to be manufactured from ADP and a phosphate. And this is going to use energy supplies by various decomposition reactions. So this is going to be particularly important. Especially the decomposition reactions, particularly that of glucose. All right. So glucose is going to be a major production factor for ATP. All right. So all of that is very important for basic energy production in the cell, in the body. Right, and that is all we have with chapter 2 with regards to chemistry, to the chemistry in the body. All right, so if there are any questions, comments, or concerns, please feel free to email me. And again, I have included some of the Wiley Plus links in the following slides. So, happy studying.